Welcome everybody to first DNAqua International Conference. So there have already been, just looking at the participants, there are over 600 participants at the moment in the conference. It's a great pleasure to introduce all of you to this meeting. My name is Florian Leese, I'm chair of DNAqua-Net, and I think not all of you are aware of what DNAqua-Net actually is or where it came from, so I'd like to briefly summarize what the origin of DNAqua-Net actually is. It's probably, simply speaking, the European version of the obvious back in 2015-16. What was obvious? Obvious was and still is that we can provide very important information on aquatic bioassessment and monitoring using environmental DNA, so molecules extracted from the water, or using DNA to validate taxonomy of specimens collected from the water. So this was obvious back 10 years ago already, and even beforehand. On other continents, initiatives have formed. Very famous is the initiative from Canada, from Donald Baird, Medad Hajibabe, Biomonitoring 2.0. Well, and we like the idea to think about, can those genetic methods be of value for monitoring in the context of European environmental frameworks? So we formed a team. Back in 2015, we wrote a proposal and applied for funds. In Europe, there is this funding scheme called COST, European Cooperation in Science and Technology, that allows to form networks around a certain topic. We formed a team, we applied for funds, and actually we made it. So our memorandum of understanding shows our main goal. The main goal is to advance the application of DNA-based tools for biodiversity assessments, and to develop a roadmap to include these in standardized bioassessments of aquatic ecosystems in Europe and beyond. The beyond aspect is very important to us. From the start, this has not been a European Union or only European effort. We have international observers. At the moment, we are people from 49 countries, and we have exchanges to all continents. I think it's really important to mention that we think this really big, we think this global. In terms of funds, it's a bit sad, or I'm not sure if we should talk about money in detail. Maybe we should. So we only have 7.2% of the costs that Cristiano Ronaldo paid for this car. This is not much. I mean, it's still a lot, 800,000 euros roughly, but this for a network of 400 to 600 people from 49 countries. This is not much for 4 years. But instead of complaining, we just accepted the challenge and tried to make the best with the money. We reached out to experts from the applied sector and from academia and formed a strong network. You see us on the kickoff here in Germany 2017 from DNAqua-Net. How is our network organized? We thought about making the network or organizing the network around 5 working groups. So looking at this figure, you will see that we have working group 3 in the upper left. So that means working group 3 is the working group that deals with the field and lab protocols, asking how consistent are results produced from different methods to actually get the data. Working group 4 deals with data analysis, so the bioinformatic processing, O2s, ASVs, taxonomic assignment. And working group 2 looks at the biotic indices. Are the indices we can derive from genetic data, are they compatible with the indices currently used for environmental monitoring or not? And very important, what else can we get out of that? Because I think with environmental DNA and other genetic approaches, we can unlock a whole new set of indicators informing about the status of our biodiversity and ecosystems. At the bottom of the working group are working group 1 and working group 5. Working group 1 deals with the question, how fit are the reference databases for taxonomic assignment for our purpose of environmental monitoring? And working group 5 asks the question, what are the barriers actually to use now these new information as part of, for example, European directives such as the Water Framework Directive or Marine Strategy Framework Directive. This is important because often there are clear standards, clear guidelines about how to assess the status. And is it compatible? Is it not compatible if we now use genetic data? Has to be addressed at this level. As I said, it's difficult to count the members because we have 5 working groups that also have their own working group lists, but between 400 and 600 members from 49 countries. We have published over 100 publications and stakeholder reports. We had 51 exchanges of individuals between labs for a couple of days to a couple of months. Then over 50 meetings and roundtables. Just as an example, on the 12th, so later this week, we will have a workshop in 15 countries. In this workshop, we will specifically discuss with the end users, in particular from federal environmental agencies, what their needs are, what their concerns are, which chances they do see in genetic methods in order to move forward with the roadmap of using DNA-based methods as part of formal environmental management programs. Let me get some details out regarding the scientific aspects. So here are some of the major papers produced by the working groups. So as I said, it's more than 100 papers, so I can only show you briefly some of the working group 1, 5, 2, 3 papers that come out or are about to come out. I think it's important. This is really the work of individuals that worked hard, not only the ones shown here, there are many more. And that's what makes DNAqua-Net really strong, that we have such a good team of people that like to work together. Also, a new journal was founded during the kickoff of DNAqua-Net in 2017. So Pensoft and DNAqua-Net realized that there's actually a need to provide a new journal, a scholarly journal to the market. This is Metabarcoding Metagenomics. I act as editor-in-chief. Dirk Steinke from Canada is co-editor-in-chief. And yeah, what's important for you maybe, there's free open access publication possible until the end of 2021. Then we will introduce a moderate open article processing charge. But I think the really important success, or the success of DNAqua-Net, goes far beyond the academic success. We tried to bridge the science application gap with DNAqua-Net. We tried to test how does it actually work, how can we implement the tools in real-world monitoring programs. Participated, for example, in the largest river monitoring activity that exists from the Danube. So 15 countries involved in the joint Danube survey, and we participated in that, taking care of genetic assessments. There have been validation studies across several nations, and there have also been, quantification approaches, how the reliability of different single species assays is. This was done led mainly by Bettina Tallinger and Kat Bruce on the right-hand side. I think what makes DNAqua-Net successful is that we like working together, and this is what we also wish to do in the future. This is not about whether old or traditional approaches are bad and new ones are good. This is all about how can we sustainably manage our aquatic ecosystem and protect biodiversity. And then it's not so much about old and new and good and bad. It is how can we consistently work together to identify for which questions which method provides the best answer. This is what we are here for. And I'm really happy that you attend the conference now. Let's now go to the scientific details. Have a look where different research institutes are right now, which new developments are coming to the market. And I hope you will stay in contact with us. Let's move environmental DNA research for the next decade so that also managers can make use of it in order to protect our aquatic ecosystems. Enjoy the conference.